Good afternoon to all. I'm delighted to welcome you all to Translation Longevity Series presentations. And in this series, Vijay will be presenting on inherited components of longevity. And our speaker, Vijay, he is a senior scientist at Renix, and he leads bioinformatics, genomics, and next generation sequencing research related activities. He is currently working on the neoantigen cancer vaccine pipeline projects. He holds a PhD in life sciences from TMC at Homi Bhabha National Institute, Navi Mumbai. His PhD study focused on cancer genomics. Previously, Vijay has worked as assistant professor at graduate and postgraduate degree colleges as well. At Renix, he has gained experience in scientific, operational, and administrative matters. So, without further delay, I invite Vijay to take over the session.
Thank you, Dr. Gita, for the introduction. Hello, everyone. Today in our Translational Longevity Series CellTalk, I will be focusing on inherited components of longevity. For this presentation, I have selected these research and review articles. So, as the topic is genetic basis of longevity, we all know longevity is the long age and healthy health span. That is the increased life expectancy of an individual. So it has multiple factors that can impact the longevity of a human, and genetic basis is one of the important factors. So the genome the individual is born with, it is a hardware which has encoded all the information that is required for the entire lifespan of that individual. And epigenome, it is the chemical modifications to the DNA and associated proteins, the way these proteins are expressed, when and where these proteins are expressed, that particular genetic control, that is the epigenome. It is the software which is influencing the gene expression and affecting the code that is contained in the genome. In introduction, so there are several factors that affect longevity of an individual, and this could be environmental factors, this could be nutritional factors, disease and disorders, and immunity associated with resistance to those diseases, and genetic factors and epigenetic factors. So today the focus is on the genetic factors, and we'll see how what genetic factors could be, how they are identified, and what impact that they can have, and the molecular mechanisms that have been uncovered, which can potentially impact the longevity or health span of an individual. So this is a theory proposed by Sebastiani in 2012. This is the hypothesis for prevalence of disease and longevity associated variants with increasing age at very old ages. So here there are three groups in which one group undergoing premature death, one group with normal life expectancy, and another rare group with exceptional longevity. So what could be the differences with respect to genetic factors in these different groups? So in one group, which is called as septuagenarian controls, there the frequency of disease predisposing variants, the variants, that is the nucleotide variants that are inherited from parents to offspring. So these inherited variants, their frequency is more, that means more such variants are accumulated in that individual, and those individuals may be susceptible to premature death. While on the other extreme, where the population is oldest of the old, life expectancy that can be achieved, there more longevity enhancing variants could be the causative factor, and these are inherited to those individuals from their parents. So if this is the genetic difference between these two extremes, then we can identify which these particular variants are, which are promoting longevity, and which variants or disease predisposing variants that are leading to premature death. And genetic analysis and genetic studies in case control manner and study of different age groups and individuals with different life expectancy, that enables us to identify these longevity enhancing variants, and thereby we can identify genes that can have positive influence on the life expectancy. So here's two examples. There are many genes that have been shown to have an association with longevity, and these are the candidate longevity genes that have been consistently replicated in multiple studies. The problem with these different genes has been that one particular study finds a strong association with longevity, but other studies do not replicate that particular finding. But these two are exception that multiple studies are reported then to have association with longevity. So one of them is APOE, and the cluster is TMM40 APOE APOC1 cluster, and another gene is FOXO3, which has been repeatedly shown to be associated with longevity. And both these genes have found the p-value less than 5 to 10⁻⁸ threshold for genome-wide significance in large jivas. So jivas are the genome-wide association studies where the attempt is to identify genetic variants, the alleles or particular nucleotides that are present in a particular location in the genome. Those particular alleles, whether they are associated with particular trait in an individual or not. And such studies are carried out in very large populations. The subject cohort is very large, and many times these are case and control cohort. And some kind of studies can be also in sibling pairs, where they find how the genome linkages of these particular traits are there. So as we have seen in earlier example, if there are two extremes in between the normal individuals or the normal control individuals, such kind of jiva studies in which exceptionally long-lived individuals they are studied as compared to other individuals. They tend to show certain alleles present at a higher frequency, which can lead to extreme longevity, and thereby we can make an association between that particular genetic trait with the longevity. So here is one of an example of jiva study in which they have identified a particular allele. So these kinds of plots, these are called as Manhattan plots, and here you can see the certain points. So these are higher p-values of association with a particular trait, and this allele, its code is RS429358, and it is located in the APOE epsilon4 gene. And this has been found to be associated with larger age group. So this APOE gene, this is a short form from apolipoprotein E, and this is a gene which is significant in the neuronal cells. And this APOE gene, it has three alleles in population, which are major alleles, epsilon2, epsilon3, and epsilon4. And these alleles are defined by combinations of two SNPs. So these SNPs are RS7412 and RS429358. These are just the designations for the identification of these particular alleles, whether particular nucleotide is present in that particular location or not. So even if they have found this particular APOE related SNPs to be associated with longevity, they have found certain differences also with respect to which allele combination is present or which SNP combination is present. So epsilon4 combination of APOE is associated with a substantially decreased odds for extreme longevity. That means these people tend to die early due to increased risk for death. And epsilon4 allele is also associated with increased risk for Alzheimer's disease. But as compared to that, epsilon to epsilon 2 and epsilon to epsilon 3 genotype, it is associated with significantly increased odds to reach extreme longevity. That means these two allele variants are associated with extreme longevity and decreased risk for death. Another gene that has been significantly linked to longevity is FOXO3. FOXO3 gene, it encodes a transition factor which may have strong impact on aging and age-related phenotypes. FOXO3, it is involved in regulation of stress response and which is theorized to affect the lifespan. FOXO3 genetic variants have been associated with decreased risks of age-related diseases and longevity. Many jiva studies have reported this. There is particular variant SNP in FOXO3, that is RS28022292, which is called a G allele, and this has been shown to have protective effects on several age-related diseases. This G allele, it has been associated with low prevalence of coronary artery disease and cancer, and fewer bone fractures, and lower cardiovascular disease incidence. This is the genomic location of FOXO3 gene on chromosome 6. At this particular location, as you can see, there are many SNPs are there, which can occur in different frequencies in the population. Out of that, this particular allele, of which the G allele, which is named as such because of the presence of guanine G in this particular genomic location. And this G allele, it has been shown to create a binding site for HSF1. And due to this binding site, this chromatin loop formation is possible. And due to that, it gains an enhancer function, and therefore FOXO3 gene expression is activated. So whenever there is a stress condition or stress situation, FOXO3 gene can get expressed. And as we have seen earlier, that this gene have been shown to have protective effects on several age-related diseases, and due to their stress response activity, and which is theorized to affect lifespan. So as we can see that Jiva studies has identified a particular gene SNP that is significantly associated with longevity, and molecular studies, like actually how this particular SNP might be affecting longevity, they have also identified the possible mechanism through which this particular gene allele can affect the longevity. In a similar way, longevity Jiva studies have identified many genes that are affecting the longevity. So these genes are ANK, RD20, A9P, many more genes are there which have different functions in autoimmune disease and blood traits. Then there are many linear non-coding RNAs, long non-coding RNAs. Then there is suppressor genes for CDKN2A. Then there are genes associated with diabetes, cancer, and embryonic development, etc. These are also some genes which have been found in genome-wide association studies to be significantly associated with longevity. Here, these are the genes, and these are the SNPs that are found to be present in these particular genes, which are having a significant association with longevity. So these are the different studies in which they have identified the significant association of these genes with the longevity. In particular, this is a set of genes, FOXO3 and APOE, which has been highly replicated as candidate genes which are associated with longevity. So in these kinds of studies, to identify, even though we know a lot of genes are associated, so majority of these studies have been carried out by using SNP arrays, which might have limited utility. As we are living in the age of genomics, so whole genome sequencing is possible, and also population level whole genome sequencing will also be possible. So in coming days, it is estimated that we will uncover more and more genes in more and more granularity of our understanding in how those particular genes are affecting the longevity. So in the present challenges that to be addressed include, in humans, the identification of underlying genes contributing to exceptional longevity and healthy aging has not been found. So strong cause and effect association has not been given because we lack the way to replicate these studies and replicate the associations that are found in multiple studies. So that will be elevated as we will get more candidate genes that are strongly associated or contributing to the longevity in individuals. And that will be satisfied when large cohorts of large-lived individuals will be recruited. But one problem with that is the population prevalence of extremely old people is low. As we have seen, the longevity is increasing as the development is happening across the world in the developed world. As longevity is increasing, so we can expect that more and more people will reach long age groups, and large cohorts of such individuals can be recruited in whole genome analysis cohorts. The replication of candidate genes significance in multiple studies is lacking. Then understanding genetic variants in the non-coding genome is also lacking, because non-coding genome is vast. More than 98% of the human genome is classified as non-coding genome, of which we know little as what function it carries out. But as it is a very large amount of the genome, it must have certain mysteries in store for us, which can include longevity or the effect on longevity. So that is one challenge to understand the non-coding genome, and the interpretation of vast volumes of genetic data that will continue to emerge. In a clinical setting, we are seeing that large amount of genetic data in terms of genome sequencing is coming up. So analysis and interpretation of that data will be challenging to associate longevity with particular gene alleles. So finally, in summary, aging and longevity has a strong genetic component. We have seen two genes, APOE and FOXO3 examples that have been shown to be associated with longevity in multiple genome-wide association studies. And large cohorts of extremely large individuals is needed to identify genes that play a significant role in longevity and healthy aging. The study of molecular mechanisms of the identified longevity-related genetic variants and related genes will help in devising longevity intervention strategies. That could be drug therapies or lifestyle improvement or lifestyle changes. The genome-level sequencing data of large populations will help to uncover more genes associated with longevity and make personal longevity predictions possible. So with this, I conclude my CellTalk on the longevity genetic components. Thank you.
Thank you, Vijay, for your insightful presentation. Now audience can ask question. Actually, I don't see any question in the chat box. So we can wind up today's session. So thank you all for your participation, and I hope you found today's presentation informative. Thank you all.